This video starts off our genetic talk, and there is a general principle in genetics, and that's called the central dogma, or the central principle. That is that DNA becomes RNA, which becomes protein. That's the central principle, the central dogma, and that will be our structure for the next coming videos. We'll start with DNA. Now DNA is long as all get out, and to fit it into something as small as our cells, we're going to really need to condense it. So if we condense DNA, we get something called chromatin, but that's not good enough. Instead, we have a protein, actually 8 of them combined, and they form kind of a bead-like structure. This is your histone. And we wrap chromatin around it just to condense it even further. This is now called your nucleosomes. How does that chromatin stay stuck on the histone? Well, DNA is negatively charged, and histones, these proteins, are made up of positively charged amino acids. These amino acids include lysine, arginine, and because of that, they can stick together. We can also clamp it down with another protein called histone 1, or H1. That ensures it gets stuck on. And by doing this over and over and over, we can really condense down that DNA, fit it into our cell. So your cell has the nicely condensed DNA. And we talked about the cell cycle last video. In the cell cycle, for things like the G phase, you're not really using the DNA, so that DNA stays nice and condensed. It is inactive. We sometimes call that heterochromatin. Again, chromatin is a condensed form, so heterochromatin. That means it is condensed, inactive, can't be reached. So H, you can think of it as H means highly condensed. However, for phases like S phase, where we do need to access our DNA, we do need to synthesize it, we need it to be opened. We need it to be nice and open. That is called euchromatin. Euchromatin is open, accessible, able to be accessed. So, accessible. What closes and opens DNA? So what opens and closes DNA in the first place? Well, there are a few methods to do that. One of them is through histone acetylation. And the names always give it away. You basically acetylate the histone. You put an acetyl group, an enzyme puts an acetyl group on the histone, lowers that charge, and relaxes the histone, relaxes the winding of the chromatin, opens it up. So opens it up, makes it accessible. That's one way. Now what if we don't want to make it accessible? What if we want to close it for things like G phase? What if we want to make it condensed and inaccessible? Well, we have histone methylation. That puts a methyl group on the histone. And to be perfectly fair, methylation both opens and closes it depending on the site, but for your intents and purposes, anytime you see methylation, that closes and condenses and makes it inaccessible. So closes. Every time you see methylation, for your intents and purposes, methylation means you condense and you mute, and for mute, the DNA. One last thing, CPG islands. Methylation Judging by the name, you methylate CpG islands. What are CpG islands? It's just areas of the DNA that are full of cytosine and guanine. That's why I call it CpG. And some CpG islands can be near transcription sites. So when you methylate it, you mute it. Again, that just closes or mutes the DNA. Hope that makes sense. That is our first topic. Now let's move on to DNA replication proper. DNA replication has many steps and is different between prokaryotes and eukaryotes like us. For example, if you have strands of DNA, prokaryotes can only have one site of replication. They can only start here. We, on the other hand, can start numerous sites at the same time. So that's one of the differences. And I'll talk a little bit more about the other differences as we move forward. But we'll start from the beginning, the first step in DNA replication. Let's start with just our DNA. So 2 strands. We need an origin of replication. Where are we going to start? We'll just say here. Now the origin of replication is usually sites that are high in A to T pairs. Why is that? Well, A and T match, and also G and C match in pairs. That's just nucleic acid pairing. G and C are triple bonded, while A and T are double bonded. We want more double bonded because those are easy to break apart. That's why the sites of replication are usually near A and T pairs, because they're only double bonded, they're easier to break apart. That's just a side note. That has appeared on questions before though, so make sure you know that A and T sites are usually where sites of replication start. So our site of replication, next an enzyme comes in called helicase, and that unwinds our double helix. Now it looks something like this. And while this opens up the double helix, sometimes it can snap back shut and reattach. We don't want that, so we have something called single strand DNA proteins. These are proteins that have a high affinity for single strands. So they attach on and make sure it doesn't reattach or anneal. That's your second step. Third step, as you're opening up the strand, you create a lot of tension. If you ever had tangled headphones and you pull on each headphone, it just makes the knot worse. And that's the case in here. If you pull on this and open up the DNA, it just makes a lot of tension and makes it worse. So we have to release that tension. We do that by an enzyme called topoisomerase, also known as gyrase. And these snip the strands, release some of that tension. Topo 1 cuts one strand. Topo 2 cuts both. Clinically, they have an important role because anti-cancer drugs can target these, like toposide. So toposide targets this, stops cell replicating. That's why they're anti-cancer drugs. Or you can stop bacterial replication by targeting bacterial toposides. Those are drugs like your fluoroquinolones. Those target bacterial topos like topo 2 and 4. So clinically they have some importance, but know in the biochem sense what they do, they snip the strands, release some of that tension. That's your topoisomerase. Now that the strands are open and the tension isn't there, we can start to replicate. A small RNA primer comes on, RNA primer. I had a question saying how is RNA used in DNA replication, and I had no idea what they're getting at. What they're getting at is RNA is seen in the primer. So RNA primer comes in, that allows DNA polymerase to bind and start making DNA. Here's where things get a little tricky. If you look at DNA's structure, then it looks something like this. Where you have a phosphate group on the 5' end and a hydroxyl group on the 3' end. When we add more nucleic acids, we use that phosphate group on the 5' end to release the energy needed to make that bond. And so making DNA goes from the 5' end to the 3' end, always. That means this strand, if we say this strand is 5 to 3, then its complementary strand will be 5 to 3 on the other end because it's complementary. This is where it gets tricky. I said we can only make DNA via the 5 to 3 direction. So let's take the top strand for example. A complementary strand would be 5 to 3, and that works perfectly for us. As we keep opening up these strands, we can continuously make new DNA. However, on the opposite end, on the other strand, we have to make DNA this way. And so it'll open up, we'll make DNA, it'll open up more, we'll have to make another piece, it'll open up more, we'll have to make another piece, and another, and another. So it comes out somewhat fragmented. The fragmented strand is going to be your lagging strand because it's always waiting for that next piece. It's always lagging behind. This, however, is going to be your leading strand. It doesn't need to wait. It can continuously make DNA. We don't want these fragments here. We want to combine them together. And the thing that combines them together is going to be your ligase. So we made our strands. Now we want to proofread. Well, DNA polymerase will not only create new DNA, but it will also proofread. So DNA polymerase 3 proofreads in a 3 to 5 direction via 3 to 5 exonuclease. So a proofread from a 3 to 5 direction, see that its new nucleotide is correct. DNA polymerase 1 is special. It proofreads in a 5 to 3 direction. And also it removes the RNA primer. We finally replicated our new DNA. But there's one more step seen in eukaryotes that we do. And it's very important. And that is, last step, an enzyme called telomerase comes in. And telomerase adds some DNA to the 3' end, just to make sure nothing got lost in the process. Because if something got lost in the process, then DNA will continuously get shorter and shorter and shorter every time it replicated. And there's a theory that when DNA gets too short, then the cell can die. And that's why telomerase is important. Now that we know what happens normally, we want to focus on what can go wrong. Mutations while you're making that DNA. So let's talk some mutations. The first one we're going to talk about is point mutations, where you add the wrong nucleic acid. And this could be misplacing a purine with another purine. that is called transition point mutation. That also includes pyrimidines to pyrimidines. However, if you put a purine to pyrimidine, or vice versa, those are called transverse. All right? Those are your point mutations. Those are the wrong nucleic acid. Kind of a subcategory of it will be our next one, which is silent. When you add the wrong nucleotide, but fortunately for us, it codes for the same amino acid. How is that so? If you've ever seen a chart of amino acids and their nucleotides, you'll notice there's some redundancy. For example, if you take valine, valine is coded by GUU, GUC, GUA, and so there's some redundancy. What is important, however, is the first two letters, the first two nucleotides. So first two important, but the third could be different, but still code for the same amino acid. First two important. Next is going to be your missense, where you add the wrong nucleotide, but this time it's for a different amino acid. This is seen in things like sickle cell, where you turn glutamine into valine. So just that one base, that one nucleotide that's different, can cause things like sickle cell. Very important. Next on our list is your nonsense mutation. This is when you've had that wrong nucleotide, and instead of making the same amino acid or a different amino acid, you actually made a stop codon. And whatever protein it codes for usually comes out truncated, non-functional. So non-functional protein. And last but not least is going to be your frameshift. This is not part of the point mutation subcategory, but frameshift is when you insert or delete a nucleotide. And that insertion or deletion causes misreading for all the nucleotides downstream. This is the most devastating. Frameshift is the worst. If we had to categorize how severe these mutations are, frameshift would be number one, then nonsense, missense, and by a far margin, silent would be the least severe, because silent is basically, there's no problem, you're making the same amino acids. So frameshift is the most severe, but know all of them. Know all the different mutations, know them well. We talked about one of the ways we can look for mutations via DNA polymerase. Our body has other methods you should know as well. One of these is nucleotide excision repair. So we take our two strands, we'll say these are the nucleotides or the bases, and one of them has gone wrong. In nucleotide excision repair, you excise the wrong nucleotide. So the endonuclease comes in, excises, and then DNA polymerase and ligase will fill it up and turn it what it needs to be. This generally occurs in your G1 phase. You look for any errors that might happen, so you don't go into your S phase with mutations. That's why it occurs in your G1 phase. You want to make absolutely sure there are no mutations before you go into your S phase. That is nucleotide excision repair. And you should know that it is defective in a disorder called defective in xeroderma pigmentosum. Xeroderma pigmentosum occurs when you have a defect in nucleotide excision repair. UV light from the sun can hit pyrimidines and cause them to link together. These are pyrimidine dimers. Pyrimidine dimers. And if you have a defect in this, you can't take that out. And so they build up and they build up, and you have a lot of mutations from sunlight. So they have to avoid the sun, or else they're predisposed to skin cancer. That's nucleotide excision repair. Our second one is mismatch repair. If, for example, these two bases were not matched correctly, then we would excise it and repair it. This occurs at G2. So right after S phase, right after you've made your new DNA, we want to make sure they're all matched correctly. So after S phase. Understand? Now, can something go wrong here? Sure. Defects in your mismatch repair causes HNPCC, or hereditary non-polyposis colorectal cancer. Patients are predisposed to colon cancer. One more, and this one is called base excision repair. These two really centered around S phase, making sure you have no mutations entering S phase, and then after S phase, making sure you had no problems making your new DNA. However, mutations can occur throughout the cell cycle, and for those purposes, we have base excision repair. This occurs anytime, and base excision repairs are a little more complex. So let's say this is our mutated base. The first step is that the base is removed. Base removed by glycosylase. And glycosylase doesn't replace it, doesn't add a new base. You actually have an empty site. We call that an apyrinic or a pyrimidic site, AP site, a site without a purine or pyrimidine. This attracts AP endonuclease and AP lyase. They see this empty site and they cut it out. The 5N is cut out by your AP endonuclease. The 3N is cut out by your lyase. And then finally, DNA polymerase and ligase comes in, fills it up, repairs it. That is your base excision repair. That does it for this video. See you next time.